All right, biology kids, let's get ready for tomorrow's mitosis and meiosis test with some vocabulary. So chromatin, remember that it's chromathin. Looks like noodles. It's uncondensed DNA in the nucleus, and that occurs during interphase. Chromatid is D for dense. It's going to be coiled up, and it makes up half a chromosome. A chromosome, since it's the longest word, it's the biggest structure. It's two chromatids that are joined at the centromere, and we usually look at those and see that they look like little X's of some sort. The centromere is the point at which the chromosomes join at their centers. It's kind of the point that they pinch together. The centriole, think about it like a pole. It establishes the north and south ends of the cell when dividing, and that's where all of your chromosomes are going to get pulled to as well. Now in meiosis, we have some new vocab as well. Sister chromatids are duplicates of a chromosome that you got from dad. So, for example, dad got chromosome 3, and then in interphase he doubles it. So the two duplicate copies of chromosome 3 are called sister chromatids at that stage. You do the same thing with mom. When you put the two together, you have 2 from dad, 2 from mom. That's a total of 4, so we call that a tetrad. Diploid refers to the number of chromosomes in body cells. It means that you have 2 copies of every chromosome. In humans, the diploid number is 46. Haploid refers to 1 copy of every chromosome. That's what we have in our sex cells. You have 23 chromosomes in sperm and egg cells. You okay, buddy? Yeah? Okay. All right, so what are the 4 stages of the cell cycle and what happens in each? As you see in the little circle diagram, we have M, G1, S, and G2. M is mitosis. That's interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. G1 is the first stage of growth where cells are just living life, doing their thing. The S phase is when they replicate their DNA or synthesize a second copy of DNA. And the G2 phase, they're just growing one last time, doing any kind of error checking on their DNA to make sure that everything's safe to move on to go through mitosis again. In any case in which the cell checkpoints at each of these stages, so at the end of G1, at the end of S, G2, etc., if there's any errors going on, the cell should die there. But if the cell checkpoints are ignored or turned off, then cancer cells can form and tumors can grow like crazy.